Welcome to Lecture 2 on the electron transport chain and oxidative phosphorylation. And to get started, let's talk a little bit about how electron transfer energy is stored in a proton gradient, the Mitchell chemiosmotic hypothesis. So in 1961, Peter Mitchell, a biochemist from Britain, proposed that energy stored in a proton gradient across the inner mitochondrial membrane, which was formed by electron transport, is what drives the synthesis of ATP. And this proposal became known as the Mitchell chemiosmotic hypothesis. In this hypothesis, protons are driven across the membrane from the matrix to the intermembrane space in cytosol by events in electron transport. So if we are looking at this from, a membrane point of view, where we have, that is going a little slow. Let me see if I can speed that up a little bit. Where we have two, I wonder if I can get rid of that. Okay, I think I've figured out my issue here. Let's see if we can make this happen a little bit more smoothly. That's better. So we're going to draw out two membranes where we have separating an intermembrane space. So this will be our intermembrane space. And then on the outside we'll have our cytosol. And on the inside we'll have our matrix. And along this inner membrane, we'll have our complex 1, our complex 2, our complex 3, our complex 4 here, and we'll also and these will be pushing protons from the matrix side. So if I label these 1, 2, 3, 4, 2 doesn't participate in pushing protons, just producing ubiquinone. However, 1, 3, and 4 do. And what we end up with is a surplus of protons in our intermembrane space. I'm going to situate this with a lot of proton density. And this results in a high pH, a high concentration of protons, which results in a low pH because pH equals the negative log of the concentration of protons. So if something's more acidic, it has a low pH. And then on the matrix side, we have a low concentration of protons and a higher pH. So it's this electron transport driven proton pumping that creates a pH gradient and an electrical gradient across the inner membrane, both of which tend to attract protons back into the matrix from the cytoplasm. Flow of protons down this electrochemical gradient, an energetically favorable process, is what drives the synthesis of ATP. And that takes place through a very specialized protein ATP synthase. And ATP synthase, which we're going to get into in greater detail in a second, has a rotor shaft, a main body, and a strator. And we're going to get into all these components. But what's really important is that these high concentration of protons physically exits out of the intermembrane space, returning to the matrix side by flowing through this protein, and as it does that, it causes a physical turning to the protein, which causes conformational changes which converts ADP to ATP through physical motion. Now, what we should do before we get into more detail with this is address a couple important questions. Firstly, what is the proton to electron ratio? So this has been an object of a lot of interest for a long time. And what this is the ratio of protons transported per pair of electrons passed through the chain. And it's really difficult to determine, and what we're going to talk about here is just really consensus of many studies. But the consensus points to being about, and this is always subject to revision, but for the sake of where we are right now, about 10 protons pumped per 2 electrons passed through the electron transport chain. And once we've established this gradient, I'm not going to go through the entire derivation and proof. However, the amount of energy in Gibbs free energy, or the change in Gibbs free energy for the flow of protons back across the from the inner membrane space to the matrix, is delta G of about negative 23.3 kilojoules. So it's a highly favorable process. So how does a proton gradient drive the synthesis of ATP? Well, without going through the entire history of it, the answer really lies in a mitochondrial complex called ATP synthase, which I've illustrated here. I took the time to illustrate it ahead because I don't think anyone wanted to watch me draw it versus just observe it before we walked through. So sometimes this is referred to as F1, F0 ATPase because it really has two portions that we'll talk, the F1 and F0 portion. The F1 portion of the ATP synthase was first identified in early electron micrographs of the mitochondrial preparations, and when they purified particles were shown to catalyze ATP hydrolysis, the reverse reaction of ATP synthase, they did some controlled experiments which showed that if they stripped these particles, the membranes could still carry out electron transfer, but they couldn't synthesize ATP. Meaning, when they stripped this protein from the membranes, they still established proton gradient. They were still undergoing the electron transport chain. However, none of that harnessed transport got converted into ATP. And when they added these back in, they were able to recover the synthesis of ATP in the system. So, getting into these subunits, ATP synthase is composed of, that same issue again, where there are F1 and F0 subunits. So, ATP synthase is actually quite a remarkable molecular machine. You can actually think of these as chemical machines. It's an enzyme, it's a proton pump, and it's a physically rotating molecular motor. Nearly all the ATP that fuels our cellular processes is made by this multifaceted molecular machine. The spheres observed in those electron micrographs, which I referred to, make up the F1 units, which physically catalyze ATP synthesis. So these F1 spheres are attached to an integral membrane protein aggregate called the F0 unit. So the F1 consists of 5 polypeptide chains, alpha, beta, gamma, delta, epsilon, and that is your alpha units, beta units, gamma, delta, epsilon units. So this is our F1 in purple. Everything labeled in red with our A, B, C, and there's 10 to 15 of those subunits, and the strators, these all make up the integral membrane protein, which is our F0. A couple important terminology points. For the F0, you have the rotor, which causes a physical rotation as protons move through. And the way that works is protons move in to subunit A, and then they interact at specific points on the C components of the protein. And to escape to the opposite side, they have to physically rotate and ratchet from one to the next to the next to the next. And this causes an actual physical rotation in the rotor, which leads to a physical rotation of the rotor shaft and conformational changes in the alpha beta subunits, which we'll get into in a minute. So these alpha beta units are catalytic sites, which are responsible for making the ATP. And the catalytic sites of ATP synthase adopt 3 different conformational structures. So the F1 structure appears at first to be symmetrical hexamer of alpha beta subunits. So there's 6 of these subunits in the catalytic zone, and they alternate alpha beta. So you'd think, okay, we have these unchanging alpha beta subunits. However, it's asymmetric in several ways. The alpha and beta subunits arranged in an alternating pattern in the hexamer are similar, but they're not identical. The hexamer contains 6 ATP binding sites, each of them arranged at the interface of adjacent subunits. 3 of these, each located mostly on beta subunits, but with some residues contributed by an alpha subunit, are catalytic sites for ATP synthesis. The other 3, each located mostly in an alpha subunit, but with residues contributed by beta subunit, are non-catalytic and inactive. The non-catalytic alpha sites have similar structures, but the 3 catalytic beta sites have 3 quite different conformations. So let's take a look at what that might look like as this rotates, as they bind ATP, ADP, and phosphate, and how this might work. So look at these catalytic beta sites. Let me see if I can illustrate this adequately. The first site in the hexamer is going to be this open or L loose conformation. Then we'll have an open conformation and a tight conformation. So open, loose, and tight. And with what these are illustrating are 3 different degrees of ligand binding, where tight binds the most strongly, loose has intermediate binding, and open has very low binding. So if we initiate this discussion with ATP being bound in the tight zone, we can reversibly show that if ADP and an inorganic phosphate are introduced to the system, they can associate with the loose system. And what that would look like is this. We put the ADP and phosphate in the L binding site, and the ATP remains bound in the tight site, and there's nothing bound in the open site. And then what we can get is an adjustment to the conformation. So if we get a conformational change to these subunits, and the tight site becomes open, it will release the ATP. The L site will become tight, binding it strongly. The open site will become loose, and what will happen is the following. And of course it takes energy to commit a conformational change. We'll call this the tight. It's still binding ADP and phosphate. However, what was tight is now open, and it's going to no longer be able to house the ADP strongly, and the ADP is going to leave. And then what was open is now loose. And then what will happen catalytically is once the ADP and inorganic phosphate are bound tightly, they will be converted to ATP in the tight binding region, and then we can get another input of ADP plus phosphate into the loose, and it'll repeat. Now, the real question here, let me shrink this down so we can see the entire thing, is what's causing the conformational change? So let's elucidate that. So going back to the proton pumping action, remember that as protons flow from the periplasm to the cytoplasm, because of this electrochemical gradient in the periplasm, what has to physically occur is that the protons have to enter the A subunit, and then bind a C subunit, and then to be released on the opposite side, this ring, this rotor of C subunits, has to physically rotate. And as it physically rotates, the rotor shaft or stalk with this gamma subunit, which is asymmetric. Notice that it's asymmetric. I'm going to highlight it here. It's asymmetric. It's attached to the C subunits. So as they rotate, it rotates. But as the gamma subunit rotates, because it's asymmetric, it differentially affects the beta subunits, which have catalytic activity. So the rotation of this gamma rotor shaft subunit relative to the beta units is what drives the conformational changes from open to loose to tight, and makes each of them alternate between loosely binding ADP and phosphate, then strongly binding it and having it form ATP, and then openly binding it or non-binding it and allowing the ATP to be released into the cytoplasm. And each of the three sites can simultaneously do all three of these things in an alternating fashion. So you can see now the whole picture of how the proton pump establishes an electrochemical gradient with all of this electron transport. And then once we have this pH electrochemical gradient in the intermembrane space, to relieve that, we can drive this electrochemically motivated motor, this rotor group, which rotates the shaft, which causes alternating conformational changes in these catalytic beta subunits, which produce ATP through conformational changes. It's just an astonishing system. This naturally raises the question, how many protons are required to make ATP? It's a heck of a question, because it depends on the organism. And it actually gives us some insight into how the evolutionary ability to acquire high energy food sources can impact our ATP production. So let me explain. So the bioenergetic cost of making ATP varies widely among organisms. For instance, in prokaryotes, chloroplasts, fungi, ATP synthasers have evolved a range of C-ring sizes from 10 to 15 subunits. So notice I labeled 10 to 15 on that. Now, since each turn of this C-ring, of this rotor, produces ATP, so one turn equals 3 ATP, because a single full turn runs each catalytic site, which there are 3 of, through the entire range of loose, tight to open. So each site has the opportunity to produce 1 ATP per cycle, and there's 3 sites. So for each full turn, we can get 3 ATP. And since each turn of the C-ring produces 3 ATP, the cost of making an ATP varies from about 3.3 to 5 protons, because if we get 10 per turn or 15 per turn, we get different ratios. So take a look at that. If we have, let me see if I can get that. 10 protons in, because each single proton must interact with a C-ring to cause a ratcheting effect. Then we get 3.3 proton per ATP. 3.3 protons per ATP. And if we had, say, 15 C subunits, it would take 15 protons to complete a complete turn. You'd still get 3 ATP, so that would cost 5 protons per ATP. So you can see there can be a major energetic difference between the number of protons per ATP depending on the number of C subunits in a rotor. On the other hand, evidence indicates that all vertebrates, or most vertebrates, contain C rings with only 8 subunits. So these are lower animals. If we have 8 subunits, that means, let's see, that's 2.7 protons, I believe, or close to, per ATP. And you might think, oh, well, why on earth would you, as an organism, make a system which requires more protons per ATP? It seems natural that you would want to have ATP formed with as few protons transferred as possible. And on the surface, that seems correct. However, vertebrates can access higher energy sources of food. And because of that, we can increase the concentration of protons in the intermembrane space to a higher level. So if you can, it takes a lot of energy, remember, to establish this proton gradient. Think of this rotor as a bicycle gear. If you can't generate a lot of force with your legs, you want to gear down the bicycle, right? Excuse me, where you have a larger gear so you can multiply the force. So if you don't have a lot of electrochemical gradient, what you need more C subunits in your rotor so that it takes less force to do incremental small turns. So if you have a lower electrochemical gradient, you need to have more protons per ATP so that you don't need as much electrochemical motive force to rotate that rotor. But if you have access to energy-rich food, you can establish a more forceful, steeper electrochemical gradient in the intermembrane space, and you can more rapidly turn that rotor to make ATP more quickly, which as a feedback allows you to more efficiently make ATP, which allows you to access those high energy food sources more effectively. So it's just a matter of tuning the balance of protons per ATP produced to the ability to establish and maintain a steeper electrochemical gradient, depending on the activity of an organism and the sophistication of an organism. So this leads us to a really important final question for the chapter. How are electrons of cytosolic NADH fed into electron transport? Most of the NADH used in electron transport is produced in the mitochondrial matrix, which an appropriate site because NADH is oxidized by complex 1 on the matrix side of the inner membrane. Furthermore, the inner mitochondrial membrane is impermeable to NADH. So let me write that down. Dagnabbit. Get rid of you. So recall that NADH is produced in glycolysis by glyceraldehyde 3-phosphate dehydrogenase in the cytosol. If this NADH were not oxidized to regenerate NAD+, the glycolytic pathway would cease to function due to NAD+ limitation. So eukaryotic cells have a number of shuttle systems that harvest the electrons from cytosolic NADH for the delivery to mitochondria without actually transporting NADH across the inner membrane. And we're going to look at those two systems, which include the glycerophosphate shuttle and the malate aspartate shuttle, and compare and contrast them. So first, let's start with the glycerophosphate shuttle, which is the first mechanism by which, as organisms, we efficiently use cytosolic NADH. So in the glycerophosphate shuttle, there's two different glycerophosphate dehydrogenases. One's in the cytosol, and one's on the outer face of the mitochondrial inner membrane, and they work together to carry out the electrons from into the mitochondrial matrix. So NADH produced in cytosol transfers its electrons to dihydroxyacetone phosphate. So what this looks like is, if we have our dihydroxyacetone phosphate, it can capture the electrons from NADH plus a proton in the cytosol, producing NAD+. And remember, this NADH that's reacting in the cytosol can't directly cross into the mitochondria because of the impermeability. So we have to find some way to chemically move it across, or have this chemical energy move across. And so what happens is this NADH is going to bind the dihydroxyacetone phosphate, and this is going to produce glycerol 3-phosphate as a product. So what this is effectively doing is reducing the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Now, this metabolite is going to be reoxidized by the FAD plus dependent mitochondrial membrane enzyme to reform dihydroxyacetone phosphate and enzyme-bound FADH2. And that looks like this. So the glyceraldehyde 3-phosphate is going to interact with the enzyme in, well, let me draw this in here. So we have our membrane, inner mitochondrial membrane, and if we have flavoprotein inserted, and there's this enzyme which is FAD dependent, the glycerol 3-phosphate can become reoxidized to dihydroxyacetone phosphate by interacting with this FAD, and this is going to generate FADH2. In the inner mitochondrial membrane space. And this FADH2 can then be directly used to be passed to ubiquinone. in the electron transport chain. So fundamentally what we're doing here is we're converting an equivalent of cytosolic NADH to an available FADH2. And remember that there's less energy available in FADH2 than NADH, so we're actually stepping down the amount of energy available, but we're able to efficiently move the energy from one side of the membrane to another because of this. So as a result, the cytosolic NADH oxides via the shuttle yields only about 1.6 molecules of ATP, so the cell's paying with a potential ATP molecule for the convenience of getting cytosolic NADH into the mitochondria. So although this seems wasteful, there's a really important payoff in this. The glycerophosphate shuttle is essentially irreversible. And even when NADH levels are relatively low in NAD compared to NAD+, the cycle operates effectively. So even though it costs some energy in the end with the efficiency of the NADH compared to the efficiency of FADH2, it's fundamentally irreversible and can operate even when we have relatively low levels of NADH available. Now, let's contrast this glycerophosphate shuttle with the other prominent shuttle, the malate aspartate shuttle, which is a reversible shuttle compared to the irreversible shuttle. Now, let's draw this out as best as we can. Divide this right down the middle, where we have our mitochondrial membrane, and we're going to have two carriers. We're going to have one here and one here, and we have an alpha-ketoglutarate malate carrier. And we have an aspartate glutamate carrier. So although we can't directly shuttle NADH, we can shuttle other compounds which are bringing the energy of NADH with it. So the way to look at this is, let's start with oxaloacetate, because that's really where this begins. So let me see if I can get the proportions of this right. So we've got oxaloacetate, and it's going to react with NADH, forming NAD+. So we're going to bring in this equivalent of NADH plus a proton, and this is going to capture that energy from the NADH. And what this is going to do is it's going to yield malate. Note that this is just a reduction of the carbonyl to a hydroxyl group. Now, this malate can be carried from the cytosol side to the matrix using this alpha-ketoglutarate malate shuttle. And so I'm just going to, let me label this. And we'll just call it malate here instead of redrawing it. Now the malate can then reversibly react with NAD+, releasing NADH on the other side using malate dehydrogenase enzyme. Which is reversible, and this is also malate dehydrogenase. We'll just abbreviate it, and this yields oxaloacetate. Now, the oxaloacetate doesn't just go back through on its own, it actually has to react with a glutamate to form aspartate to go back through. So it gets a little bit complicated, but we have a reaction with glutamate forming aspartate. The aspartate goes right through. And then, let me actually make a little bit of room here. And then the aspartate rebinds with alpha-ketoglutarate to form oxaloacetate and glutamate. I know, a little bit odd. I didn't leave myself quite enough room here, so. Let me redraw this for you guys. Give myself a little bit of room here. Aspartate's going to be down here. And then you're wondering, where did that alpha-ketoglutarate come from? Well, when the glutamate interacted with the aspartate and oxaloacetate, there was an offshoot of alpha-ketoglutarate, which then counter-shuttled there. So for each malate across, we get an alpha-ketoglutarate back, and for each aspartate that we get across, we get a glutamate back as part of the shuttle. Now, you're thinking, this is awfully silly. You don't have to have this completely memorized. The important aspect of this that you really need to focus on is the following. We have oxaloacetate. Oh, exit drawing. There we go. There's the red. Oxaloacetate picks up NADH on the cytosol side via malate dehydrogenase, forming malate. Malate is then actively shuttled using the alpha-ketoglutarate malate shuttle into the mitochondrial matrix. The malate can then interact with malate dehydrogenase on the matrix side, releasing the NADH. This re-establishes oxaloacetate, which then, through a couple other steps, gets pulled back through in the form of aspartate, has to react with alpha-ketoglutarate to reform oxaloacetate again. So the important thing, NADH goes into malate, NADH comes out of malate. Now, in the last shuttle, NADH became an equivalent of FADH2 and lost energy in the process, but it was irreversible and therefore able to drive under all conditions. The malate aspartate shuttle is different because we don't lose any of the energy from the NADH2. We get an NADH from the cytosol, effectively moved to the matrix via the shuttle with no loss of energy. Now the difference is this is a reversible shuttle, and it operates only if NADH to NAD plus ratios in the cytosol is higher than the ratio in the matrix. We have to have a surplus of NADH to NAD in the cytosol so that it's motivated to cross into the matrix. But when we have this condition met, because the shuttle produces NADH in the matrix, the full 2.7 ATPs per NADH are recovered. So that's a bonus. And those are really the two mechanisms by which cytosolic NADH gets transported actively into the mitochondrial matrix and can be utilized for the production of ATP. And that's also the end of this chapter for us. So on that note, I hope you enjoyed this content. I think it's really fantastic. insight into how all of these reducing equivalents, which we generate via the systems that we've been discussing throughout the semester, ultimately establish a proton gradient, which drives a sequence of sophisticated electrochemical motors, which physically change conformations of enzymes, which actively make ATP. So we have a few more chapters to go through, but this is the end of this one, so I'll be talking to you about subsequent chapters in a little bit. Bye-bye.